And then there's the part of this story that experts describe as the real, tangible gap in the current safety system, the place where oversight actually breaks down in practice rather than just in theory. Right now, when a scientist wants to bring a digital genetic sequence into physical existence, they typically send that sequence to a DNA synthesis company, a business that specializes in manufacturing custom DNA to order. Many of these companies voluntarily screen incoming orders against databases of known dangerous pathogens, flagging anything that resembles a genetic sequence associated with a known biological threat. But here's the catch that biosecurity experts are sounding the alarm about. That entire screening system was built to catch sequences that resemble already known, previously documented dangerous organisms. It was never designed to catch something that has never existed before, a sequence with no prior entry in any database anywhere, because nothing like it has ever been seen or cataloged. In other words, the existing safety net was built to catch a copy of a known threat, not an entirely novel creation with no historical fingerprint to compare it against. And crucially, this screening at DNA synthesis companies is currently voluntary in most cases, not a strict, universally enforced legal requirement. Biosecurity researcher Philippa Lensos of King's College London has pointed directly at this exact point in the process, the moment a digital design becomes physical DNA through a synthesis provider, as the most realistic and practical place to actually build stronger oversight, since it's the one clear choke point standing between an AI-generated genetic design existing only as data on a computer and it becoming an actual physical, living organism in the real world. There's also a genuinely important detail worth knowing about the underlying AI model itself. Evo2, the more advanced of the two systems used in this research, is openly available to researchers around the world, published as open source software, meaning any team with sufficient computing resources and technical expertise could potentially access and build upon this exact same underlying technology. That openness reflects the scientific community's broader commitment to transparent, reproducible research, a value that has driven enormous progress in biology and medicine for generations. But it also means the tool that made this specific breakthrough possible isn't locked away in a single restricted lab. It exists in the wild, available to the global scientific community, for better and for worse. So, should you actually be worried? Let's be honest and balanced about this, because both overreacting and dismissing the concerns entirely would be a disservice to what's actually happening here. On one hand, nothing about this specific study created anything remotely capable of harming a human being. These are bacteria-killing viruses, deliberately restricted through careful, responsible research design, published transparently through peer review specifically, so the broader scientific and biosecurity community could scrutinize the work and respond appropriately, which is exactly what's happening right now. This is, in a very real sense, science working the way it's supposed to, advancing a genuinely promising medical tool while simultaneously and openly confronting the safety questions that tool raises. On the other hand, the biosecurity experts raising concerns aren't reacting to hype or fear-mongering. They're pointing at a real, documented, and currently unresolved gap between what this technology can now technically do and what our existing oversight systems were actually built to catch. That gap is real. It's been stated plainly by credentialed experts in a peer-reviewed scientific journal, and it is not yet closed. What happens next matters enormously, and it's genuinely still being worked out in real time. Biosecurity experts are calling for the development of new detection tools specifically calibrated to identify AI-generated genetic sequences, systems built to catch things, precisely because they've never been seen before, rather than because they match something already known to be dangerous. There are also growing calls for clearer, more consistent, and less voluntary screening requirements at DNA synthesis companies, the exact choke point where a digital design becomes a physical, living reality. Whether governments, scientific institutions, and the biotechnology industry move quickly enough to close that gap before the technology advances even further is the actual open question hanging over this entire story, far more than any single headline about 16 new viruses ever could capture on its own. This is what real scientific progress often looks like up close, messier, more nuanced, and more genuinely complicated than a single dramatic headline can ever fully convey. A team of careful, transparent researchers built something with real, tangible potential to help solve one of medicine's most stubborn modern crises, publishing their work openly, specifically, so the world could see it, question it, and respond to it. And, in the very same breath, credentialed experts in the field are honestly telling us that the guardrails meant to keep this kind of capability safe simply haven't caught up yet. Both of those things are true at the exact same time, and understanding that nuance matters far more than either blind panic or blind reassurance ever could. If this kind of story matters to you, the kind that actually explains the real science behind the scary headline instead of just repeating it, hit subscribe so you don't miss what comes next, because this conversation is only just getting started. Now, before your imagination runs somewhere dangerous, Here is the single most important fact in this entire story, the one detail that should genuinely reduce your immediate alarm. These are bacteriophages. That word specifically means viruses that infect bacteria. They cannot infect humans, animals, or plants. They are biologically incapable of causing an infection in a person the way a flu virus or a coronavirus does, because they are built to recognize and attack bacterial cells specifically, not human ones. And this wasn't an accident or an oversight. The research team deliberately excluded any genetic sequences from viruses capable of infecting humans, animals, or plants from the AI's training data, specifically to keep the risk contained during this experiment. In other words, the scientists intentionally built a safety boundary directly into the project from the very beginning. And there's a genuinely exciting, deeply promising reason this research exists in the first place, one that gets buried under the scarier headlines. Bacteriophage therapy, using viruses that specifically hunt down and kill harmful bacteria, is already an active and growing area of medicine used in clinics around the world to treat bacterial infections that have become resistant to every conventional antibiotic doctors have left to try. Antibiotic resistance is one of the most serious, quietly escalating public health crises on the planet right now. A slow-motion emergency where common infections are gradually becoming untreatable because the bacteria causing them have evolved past our existing drugs. Finding new, effective bacteriophages that can outmaneuver resistant bacteria has traditionally meant scientists searching through soil samples, sewage, and natural environments, hoping to stumble across a naturally occurring virus that happens to do the job. This AI-driven approach flips that entire process. Instead of searching nature and hoping to get lucky, researchers could potentially design a custom bacteriophage on demand, engineered specifically to target a particular drug-resistant bacterial strain, dramatically accelerating the search for new treatments in exactly the crisis where doctors currently feel like they're running out of options. So that's the promising side of this story, and it's a real, legitimate, exciting one. But here's where the more serious conversation begins. And it's a conversation biosecurity experts started having the moment this paper was published, not months later. Alongside the original research, the journal Science also published a companion editorial written by biosecurity specialists from Johns Hopkins University, and their conclusion was blunt. Thomas Inglesby, director of the Johns Hopkins Center for Health Security, along with his colleague Moritz Henke, stated plainly that the ability to design a viral genome using AI now exists, and the rules for doing it safely simply do not exist yet. That sentence is the entire crux of the controversy in 9 words. The technical capability has arrived faster than the safety framework meant to govern it. Here's the core of what worries biosecurity researchers, and it's worth understanding clearly rather than just reacting to a scary headline. The research team in this specific study made a deliberate, responsible choice to exclude dangerous genetic sequences from the AI's training data. That was a conscious safety decision made by ethical, careful scientists working openly and publishing their methodology for peer review. But experts point out that this kind of self-imposed restriction is a choice, not a fixed technical limitation built permanently into how this type of AI model works. A future, more capable version of the same general technology, or a similar model trained by a different team with fewer safety precautions, or with an intentionally broader and less carefully filtered training dataset, could theoretically not be bound by that same voluntary restraint. The tool itself, the underlying genome language model approach, doesn't inherently know or care whether it's being used to help cure antibiotic-resistant infections or for something with a much darker intended purpose. The responsible guardrails that existed in this particular case were a decision made by the people running this particular project, not an unbreakable law of the technology itself. 16 viruses. That's how many new life forms scientists just brought into existence that had never once existed anywhere on this planet before. Not evolved, not mutated, not discovered in a swamp or a bat cave or a hospital ward. Written, typed into existence by an artificial intelligence the same way a chatbot writes a sentence, except instead of words, this AI was writing the actual genetic code of life itself. And then, humans took that code, built the real physical DNA from scratch, dropped it into living bacteria, and watched something that had only existed as data a few days earlier come alive and start killing. This isn't science fiction. This isn't a movie trailer. This happened. It's published in one of the most respected scientific journals on Earth. And the scientists behind it are now sitting across the table from biosecurity experts who are asking a question nobody has a clean answer to yet. Should we be worried? Stick around, because the real story here is more fascinating, more promising, and yes, more unsettling than the headlines are letting on. Let's start with exactly what happened, because the details matter enormously here, and a lot of the panic floating around online is based on people not actually understanding what these 16 new viruses are, or what they can and cannot do. On August 6th of this year, researchers at Stanford University and the Arc Institute, A nonprofit research organization focused on AI and biology published a study in the journal Science that represents the first time generative AI has ever designed complete functional viral genomes entirely from scratch. The project was led by Brian High, an assistant professor of chemical engineering at Stanford, who also serves as an innovation investigator at the Arc Institute, working alongside Stanford bioengineering graduate student Samuel King, who served as the lead author on the published paper. Here's how they actually pulled it off. The team used two AI systems called AVO1 and EVO2, which are what's known as genome language models. And that phrase is worth sitting with for a second, because it explains everything about how this technology actually works. A regular large language model, the kind that powers a chatbot, learns the patterns of human language by training on enormous amounts of written text, and then predicts what word should logically come next in a sentence. EVO does the exact same thing, except instead of training on English or any human language, it trained on the genetic code itself, the actual DNA sequences of more than 2 million real bacteriophages, a category of virus that specifically infects and kills bacteria rather than humans, animals, or plants. After learning the underlying grammar of that genetic code, the model can generate entirely new genetic sequences, the same way a language model generates a new sentence, one genetic letter predicted after another, based on everything it learned from the patterns of real, natural viruses. The researchers gave the AI a specific starting point to work from, a small, extremely well-studied natural bacteriophage called Phi-X174, one of the most thoroughly researched viruses in the history of molecular biology, chosen specifically because scientists already understand its structure and behavior in enormous detail. Using that as a kind of template, the AI generated over 300 candidate genome designs. The researchers then took those digital blueprints and turned them into actual physical DNA through a standard laboratory process called chemical synthesis, essentially manufacturing the real molecule based on the digital sequence the AI had written. That synthesized DNA was then introduced into live E. coli bacteria in laboratory dishes, and the team simply watched to see what would happen. Out of those 300 plus AI-generated designs, 16 actually worked. 16 genomes that, when placed inside real bacteria, successfully assembled themselves into fully functional living viruses, capable of replicating and killing their bacterial host exactly the way a natural virus would. And every single one of those 16 had never existed anywhere in nature before that moment. Not a variant of something that already existed, not a mutation of a known strain. Entirely novel genetic sequences, conceived by an algorithm, brought into physical existence by human hands, and functioning as real, living biological entities the moment they were introduced to their target. When researchers combined all 16 into a single cocktail and tested it against E. coli strains that had already developed resistance to a naturally occurring bacteriophage, the AI-designed viruses successfully wiped out 2 separate drug-resistant strains. In one especially striking detail uncovered afterward through cryo-electron microscopy imaging, one of these lab-made viruses was found to be carrying a DNA packaging protein that was evolutionarily distant from anything typically seen in this class of virus, a structural feature the AI seemingly arrived at on its own that didn't closely mirror what nature had already produced.